So next discuss the structure of antibodies and their formation, their structure, function. So antibody, as we know, that it's the product of plasma cell, and which are the product of B cell. So B cell gives rise to plasma cell. Plasma cell produces antibodies. So antibodies have a very unique structure. There is two heavy chains, and in these heavy chains we have constant and variable regions. We have constant 1, constant 2 and constant 3 regions, and we have two disulfide bonds, 1, 2, and these two disulfide bonds are between cysteine and proline amino acid, which makes the region of Hinge. So there are long heavy chain constant region with two disulfide Hinge region, and after this three constant and one variable region, we have another light chain. And this light chain region, there is an also disulfide bond between heavy chain and light chain, and there is so this is the C of heavy chain, C of heavy chains, constant region of heavy chain. This is the hyper variable region of heavy chain. Then we have light chain, and in light chain we have one constant and one variable region. So this is variable constant entities of light chain. So this is light chain. There is only one light chain and one light chain variable region and one constant region, but in heavy chain we have three constant regions. So the portion which is present on this side and also on this side, it's the Fab portion. And what is Fab? Fab is actually, it's a fragment of an antibody, fragment of antigen binding site, and it is encountered with a lot of epitopes, and this Fab region is determine idiotype. What does that mean? Idiotype means what antigen bind to what antibody, and this region has only, it means only one antigen is specific to one antibody per B cell. So Fab consists of variable and constant regions of both heavy and light chain. So Fab has both heavy and light chain region, and it binds only epitope or antigen or epitope. So it's an antigen bind. Fabs are smaller as compared to the Fc part of the antibody. They have the better pharmacokinetics, and they have better tissue penetration. So Fabs are used in immunotherapy therapies like clonal antibodies. So in contrast to Fab, we have an FC portion. This FC portion has 5 C's. First one is FC portion consists of only constant region, 2 constant. FC portion consists of only heavy chain. It consists of only constant regions. One part is phagocytic cell, phagocytic binding site, this one, and other part is complement binding site, and then they determine isotype. What does it mean? Like in isotype switching, only the constant region changed. That's why they determine isotype. So all the isotype have the same this function, this structure, but they have variable different variable regions. So IgA variable region is different. So but the constant part of all the Igs, all the immunoglobulins are the same. So they determine the isotype. They are not useful in because they are larger in size. Large size mean poor pharmacokinetics and poor tissue penetration. They are not useful in immunotherapy. Then we have two things: antibody diversity. So how antibodies are diverse in nature, how they are very much different from one another. So in the bone marrow, B cells form, mature, and immunoglobulins. In the bone marrow, the immunoglobulins are undergo a process of recombination, which is performed by recombination, and this recombination is by VDJ recombination or VJ, and this recombination is random. So the random recombination of VDJ rearrangement give rise to antibodies diversity. Then we have when an antibody is self autoimmune, like it is self-reacting, then there are 3 pathways. First one is it is apoptosed. Second one is it is clonally inhibited. So there is and the third one is they change the light chain. There is a gene known as recombination activating gene, which is known as RAGE gene, and this can determine this VDJ rearrangement, and this gene transform the light region of light here like VJ region to change their VJ region, or actually it is a light chain editor, and it change that self-reacting immunoglobulin light chain and then make it harmless. So if this RAGE gene is deficient, or is the RAGE gene mutated, Then we have a condition known as severe combined immunodeficiency. So after a recombination, we have an enzyme known as terminal decarboxy deoxy ribo deoxynucleotidyl deoxynucleotidyl transferase. This enzyme add nucleotides or transfer nucleotides randomly during this recombination, and leads to give the biodiversity. So this enzyme and this gene working together to give VDJ recombination rearrangement. So there is a random combination of light and heavy chain. So this all happens in the bone marrow. There is also a process known as affinity maturation, which is also happen in bone marrow, but it does not give biodiversity, it gives antibody specificity. So next thing is antibody specificity. Like antibody specificity means the antibody is specific in its action, specific in its working, and it is antigen dependent. Like whenever the immunoglobulin is antigen exposed, or B cell is antigen exposed, then it can form immunoglobulin against that antigen and also form a memory cell. So whenever there is there is another exposure, so the antigen specificity, in which there is three processes: affinity maturation, somatic hypermutation, and isotype switching. The response is very much specific in nature, which leads to a re-exposure defense system in our body. So for antigen specificity, we have first in the bone marrow affinity maturation, which means that the affinity gets matured, affinity maturation, and it leads to affinity means increase in the strength of binding of immunoglobulin to the respective antigen. So affinity maturation is actually the strength of binding strength, and it's get matured in the bone marrow. After affinity maturation, the immunoglobulin travel to the lymph node germinal center, where somatic hypermutation happens. Somatic mean there is a leading somatic mean bodies or bodily cells, and hyper mean fast or increase, and mutation mean there is a mutation in the cells which are very much fast and which give rise to the affinity antibody specificity. So this affinity maturation, somatic hypermutation, it happens in lymph node germinal center. And whenever there is an increased level of somatic hypermutation, which leads to germinal center hyperplasia, which gives rise to lymphadenopathies. After affinity maturation and somatic hypermutation, we have isotype switching. Isotype switching means now the VDJ give rise to an immunoglobulin IgM or IgD. So that's IgM or IgD come for affinity maturation, they get matured, they get hypermutate, and now they need to switched, and this is mediated by TGF beta, and it leads to conversion of IgM or IgD, IgM into IgD and IgGA, and respectively to the with respect to their exposed antigen. If there is a parasite, the IgM is converted into isotype switched into IgE. There is a response against tuberculosis, this IgM is get converted into IgG. So if there is a there is an intestinal infection, this IgM get converted into IgA. If this isotype switching sorry, if this isotype switching is impaired, then we have a condition known as hyper IgM syndrome in our immunodeficiency section. So this is all about structure and function of antibodies. We have known all these structures, all these hinge, these all are MCQs, and it's quite much hot topic for USMLE. It's a high yield topic. I have seen a lot of question, a lot of confusing question for immunology section. And what we have left here, we will discuss this isotype switching in our next immunoglobulin lecture. So for now, we have discussed T cell differentiation, T and B cell, T cell and APC's activation and structure of antibodies. Next we will discuss the types of anti-immunoglobulins and isotype switching.